expertise in the field of chemistry, biophysics, molecular biology, biotechnology, biochemical engineering, and many more. He has completed his BSc MSc from University of Calcutta, followed by an MTech from IIT Kharagpur and PhD from University of Illinois at Chicago. Dr. Hazra's academic pursuits have him taken across continents, including prestigious postdoctoral position at Dr. Jude Children's Research Hospital in USA and Albert Einstein College of Medicine in New York, USA. His illustrious career sorry. His illustrious career is further adorned with over 50 publications and multiple patents, reflecting his invaluable contribution to the scientific community. Today, Dr. Hazra will grace us with the expertise and insight as he addresses on the topic combining computational strategies and experimental validations by Hazra Group towards combating antibiotic resistance. Without further any delay, I now hand over the platform to Dr. Hazra for his enlightenment talk.
Is it visible? Is it visible?
Yes, sir, visible.
Okay. So I'm starting, okay? So first of all, thank you for the nice introduction and thanks for inviting me to this hands-on workshop on high performance computing in bioinformatics. Today, as Professor Rai was talking about the combination of work, my talk, which I try to make it in a generalized way rather than putting a lot of results, will show you how computation is actually help us strategizing the experiments, because you cannot do a lot of experiments. So using computation, you could target the broader perspective and then narrow it down to. Hello.
Saugata, please wait for a minute. So presentation is not coming in full screen. So
Okay. Okay.
did you share through Teams, Saugata? Okay. Okay. Okay.
Is it okay, or I will do it again? Should I do it again?
Wait.
Okay. Okay.
Now we can start.
Okay. So thank you again for inviting me. So as Professor Wright told, my today's talk is a kind of nice representation of how computation help us to look at in a broader perspective to the problem, narrow down the important aspects and validate through experiment. So that's why I have made the title Combining Computational Strategies and Experimental Validation by Hazra Group towards combating antibiotic resistance. To start with, antibiotic resistance, this is a very global problem nowadays. After pandemic, people are putting more like stress to even the PSA office, Principal Scientific Advisors Office of Prime Ministers. They have now announced One Health program in antimicrobial resistance. So what is antibiotic resistance? When bacteria have made some changes in their targets so that a drug which was already working, stop working now, is the salient feature of antibiotic resistance. The emergence of the antibiotic resistance against popular antibiotics significantly reduces our arsenals, what drugs we are having, and it is becoming difficult gradually to fight against the deadly pathogens. And how serious it is, you could realize this through the statement taken by World Health Organization that the threat of antibiotic resistance is no longer a prediction for the future. It is happening right now in every region of the world and has the potential to affect anyone of any age in any country. If you look at this slide, you would kind of again understand the seriousness of where antimicrobial resistance is standing now. So these are projection between 2018 and 2050, the common death causes like measles, road accident, diarrhea, diabetes, cancer, and antimicrobial resistance. For all of them, where the rate of death causes are like nearly same or even reduced, but for antimicrobial resistance, there is a projection of whooping 100-time increase. That shows how serious it is if not proper measure would be taken immediately. Without antibiotics, a simple mild wound could be life-threatening. Pregnancy and childbirth could be prohibited. Operations would be banned. Food consumption would be heavily scrutinized. And in one word, it would create a like situation which doctor says the patient is diagnosed with the disease, but there is nothing to do, which says diagnosed for death. So why antibiotic resistance is such a big problem? People say it is a war between humans and microorganisms. But in our normal knowledge, if we look at the war, we have gone through a lot of war. They are not impossible to combat with. So we talk about civil war, we talk about world war, uh, all these scenarios. So what is special about it? The war I'm talking about is a complete one-sided war, a war which we realize through our life experience. The young audience who are listening to me probably would have not experienced this, but people who are of my age, they know already what I'm talking about. It is a fight of unequals. It is a fight between husband and wife where one is unarmed, where other is, always ready and totally armed. One defend, another attacks, and we know who is the clear winner. So coming back to science, what is the science behind? The reason is nature, both human and microorganism, we are the kids of the nature, and human with their advanced technology try to kill protect, combat microorganisms, but nature, knowing microorganisms are not being existing or they could have lose their existence, always empowered them so they could make the fight. The reasons which nature have given to bacteria is extreme adaptability of the bacteria, amazingly small doubling time, error prone replication. Error prone replication is a very big reason where they could change their DNA, and one part of my talk is focusing on that part. And then the most exciting part is gene transfer mechanism. Among us, we cannot exchange any character. Like if I am strong with one character, even if I want, I cannot give that character to other. Whereas if a bacteria is empowered with a character like resistant to one drug, it could pass it on to other bacteria. With the mechanism of transformation, conjugation, and transduction, and by that way, one bacteria become resistant, the resistance spread in the whole world and make this scenario more critical. So how could we win the war? So what should be our strategy? If it would be between a wife and husband, as I told, the only way is to say sorry. But as it is not, in Hazra lab, we have developed a plan which is called DTMS model, which is diagnostics, therapeutics, mapping the resistance scenario, and regular surveillance. Unlike other diseases, when you consider antibiotic resistance, you don't apply medicine always. You first want to understand how serious the infection is, and that's why here diagnostics come more important than therapeutics. And in our lab, we did that study targeting a very critical enzyme family, which is the enzyme beta-lactamase, using interdisciplinary approach of computational and experimental techniques. So when I'm going forward to the main topic, you might wonder why I target one enzyme, beta-lactamases. Because if you look at this slide, you will see that this is the total antibiotic use in each country till September 2020. And the graph will show you two things. One, greater than 80% of the antibiotic used belongs to the beta-lactam group. And other thing is, if you look at the countries, you will not get the mention of developing or underdeveloped countries. Why? Because data inconsistency is a big thing. Because of that, we don't have proper data, though this like infection is a very big problem, even bigger problem than developed country to the developing and underdeveloped country. Also, beta-lactams being cheap and rapidly working is the mostly sold drug without prescription through over-the-counter. This is called OCT problem, over-the-counter problem. The drugs which are consumed over-the-counter directly on the medical shop are not included on the total consumption list. Overall, it is the mostly used and major antibiotic resistant enzyme, beta-lactamase producing drug. A lot of people tell me, so you are working on beta-lactamase. Is it one enzyme? Not at all. First of all, there are four distinct classes of enzymes. There are four different type of beta-lactamases, which are all individual and very big family. But in addition to that, there are enzymes which are dealing with the beta-lactam drugs are transpeptidases, metallohydrolases, penicillin recognition proteins, and penicillin binding proteins. So overall, it's a very big family of enzyme class. Today, I will focus on class A beta-lactamase so that it would be easier for you to follow me. So 9 years earlier, when I joined IIT Roorkee and developed a lab, there was a lot of questions to start with. For example, how many class A beta-lactamases are present in the nature? How similar are they? Which ones should we focus, target? What would be our approaches to study them? As I told, due to data inconsistency and all, these questions were not answered. We don't have good documents and all. So we decided to go through and develop our own database. So we study individualistic beta-lactamases, we develop criteria, and based on those criteria, we have developed a script so that we could have screened through the literature and find out the beta-lactamases which are reported not in a proper way, but at least with features which are already established in the existing literature. We just wanted to collect them, then we wanted to continue further. So our script with a series of trial and error was optimized, and we have find out more than 25,000 proteins which are beta-lactamases in the subclass of class A. Then we start refining them with difference in the gene, difference in the organisms, the bacterial target, exclude the sequence with organism not specified, excluded sequence with higher divergence like partial sequence and all, excluded point mutation variants and all. This brings us to refined 1916 sequences. And when we further analyzed them and we did some clustering, we have seen that when the enzymes are clustered, they are clustered into groups, and many of them coincide with the previously identified subclasses, like you see here, SHB, TAME, SME, which are already identified subclasses, but people have not done proper clustering or proper documentation about that, but that confirm us that we are going on the correct direction. So with that, we have proceeded further, and remember we talked about the questions we had, how many class A beta-lactamases are present. Now we know that around 150 different unique subclasses are present. How similar are they? Sequence wise, when we consider the gene sequence and protein sequence, they range from 90 to 40% identity. What I say identity means the amino acid are identical. Like if there is a glutamate, there will be a glutamate, but there is other category like very similar and similar. So we have classified them into 4 category: identical, very similar, similar, and not at all similar. Interestingly, then from the sequence we shift to the fold, and the fold is the 3D structure of a protein which defines the function of the protein. Here in the class A beta-lactamase, in spite of the identity becoming 90% or 40%, the fold are strikingly very similar. But with a little change in the catalytic efficiency, there is a possibility of alteration of the catalysis. So that means if you are being a drug designer, test it with one enzyme, you might not get the proper information. You have to find out who are the candidate which are diverse from each other, and you have to bring a set of enzymes. Which one we should focus? We need to make more individualistic and comparative study. So in this moment, we don't know how to, and that will bring us to further what could be our approaches to study them. So as I told, we got all these class A subclasses identified through our program. All these subclasses we have identified, and then we go for further, we go to particular study. So what we did, Among these 150 plus beta-lactamase subclasses, they all have natural variant. What that means? So you target an enzyme and nature change the chemistry of the amino acid of those enzymes so that their catalytic property altered. So we have picked up many such classes. Here is an example of the workflow where we have taken the enzyme CTXM. CTXM have 428 variants. So there is the wild type, the original enzyme, and 427 different natural mutants which is made by nature, identified by us from the literature through our program. So first we have collected all those sequences. Then we go for sequence alignment to compare them through multiple sequence analysis, calculation of mutational frequency and all for each of the positions. Then we go for 3D structure. We go for structural modeling of the all 428 sequences. We have automate the process of energy minimization and creation of analysis, which is Ramachandran plot and energy. Once we have the 3D structures, we have performed molecular docking. So for each of these variants, like 428 variants, we have used 7 individualistic chemistry of drugs, which means we have performed 2,996 docking only for the CTXM subclasses. Once we have the docking, we have done the analysis, we have picked up few of the important variations, we have gone from molecular dynamic simulation, which is also automated. So now you might think that it is a huge amount of work, and how we are working on that. Thanks to an initiative taken by our group, mainly became successful through the COVID period, where a lot of students have joined us through online. We have make protocols, we have make videos, we have make training and all to make them trained for this, and they had gone through one or like few people together with one of these sub classes. So last 8 years, 200 plus interns have worked through that, and it is incomplete without acknowledge the immense contribution of those who have now helped us creating the enormous data with us. As I told, we have developed automated docking pipeline where we have used 7 chemistry of drugs: penicillin, cephalosporin, carbapenem, MBIs, monobactams, DBOs, and boronates. So those are the scripts and all we have used, and we use the platform of Autodoc to perform this automated docking pipeline. Then we go for automation of the simulation. We are like having the protocol, and we are indated here. We have used Param Ganga. Param Ganga, the supercomputer, is based on heterogeneous and hybrid configuration of Intel Xeon Cascade-like processors and NVIDIA Tesla V100. There are 312 CPU, GPU, all those nodes with a total peak of computing capacity of 1.67 petaflop performance. The cluster consists of compute nodes connected with the Mellanox infinite band interconnect network, so people could access this from even outside the campus, and the system uses the cluster parallel file system so that you could have run them all in parallel, which helps us running those time consuming simulation in much shorter time. So we come to the multi-level computational analysis, collecting those data, where if you see all of them, BLASI, SHB, TAME, BLI, they are different enzymes but having very characteristic 3D fold. And we are looking to the active site, omega loop, SDN loop, KTG motif, SXXK motif, and towards that we are comparing the gene sequence, the protein sequence, the structure, complex, the protein and the drug, the dynamics in simulation, and ultimately the network of the mutational development. In summary, our goal is to create a comprehensive database for all variants of beta-lactamases. This project aims to create a platform for researchers to access practically all type of data on beta-lactamase. As I was showing now we have already documented the data which is coming through in silico analysis, but on the second phase, we are also including the experimental data which are available in the literature. We also anticipate that our study will serve as a toolkit for drug designers producing new anti-AMR therapies, and the data inconsistency will no longer be an issue in the case of beta-lactamase after it is completed. This will also help predict the chance of new resistant variants to emerge. As I told, nature will continuously create the changes. So if we have this data, it would be very easy for the scientists to take this data and make a pattern towards predicting the newly emerging resistance, which will be a real, like stage ahead so that we could combat this problem. We also hope that this will make the fight against AMR a little easy. So these are the different, subclasses with variants, and total variant of the class A beta-lactamase are already, we have collected 12,000 plus. This is the global beta-lactamase database which we are developing, containing all the data which will be publicly accessed within a short while. Few of them, the link is here, is already published in the website. So, okay, we have done those analysis, but what is our direct finding? So based on our analysis, we have come up with a minor group in the subclass of class A. The major subclass, we name it as A1 and the minor one as A2. So based on conserved residues and significant insertions, the class A beta-lactamase are classified into two subclasses. The major subclass were already existent, but people didn't call it as a major subclass. People consider this as a only subclass. We have come out with the minor subclass through our analysis. We got 43 conserved residues which were characterized in the
link.
Hello. Hello.
Sorry, sir,
Hello?
no problem.
I should continue?
Yes, sir.
Okay. 43 conserved residues were characterized in subclass A2, which distinguished them from subclass A1. We have done analysis where we have found insertion segments. So few patches of amino acid are inserted, which are very interesting changes. And we compare them to see the fold and the composition of omega loop. Omega loop is the most important loop for the catalysis of the enzyme. So if you see, you will see that this is having an extension here. And if you compare the omega loop, the red is the A1 and the blue is for the A2, and you will clearly see the changes. Because of this change, the minor group show extended spectrum beta-lactamase activity. The other thing which we immediately get information through our study is identification of unique insertion mutation in the KPC beta-lactamase. KPC beta-lactamase belonging to a bacteria called Klebsiella pneumoniae. This bacteria is very, very prevalent on the hospital system. 70% of the identified bacteria creating infection are Klebsiella. So if you see, it is amazing that we have like gone through the literature and identify out, especially I'm giving two examples. One is KPC 107. This is having 28 amino acid insertion, which means that there are 84 nucleotide which is inserted on the gene. And this is happening near to the omega loop, the most important loop, again having 18 amino acid. So together they form a loop region consisting of 46 amino acid. This is a loop region not generally seen in any of the common protein with a size of 300 to 400 amino acid length. Also, we got something called KPC 44, where we have seen 15 residue inserted in a 11 amino acid loop, which means the loop is now having 26 amino acids. So the insertion is an upcoming problem in the field of beta lactamase. This insertion variants, which is now in KPC, as I told, they could spread to others. So it could spread to other bacteria, and that might create a new pandemic, because with this enhancement, they are coming up with a very different catalytic activity. I will talk about little bit when I go to the experimental part. But the good part is that we are the first one to identify all of them because of our initiative to having a database. So overall, we found 4 regions, one between the alpha 3, the alpha helix 3 and the alpha 4, which is the number of leucine 102 to serine 106. Another one I already told, the omega loop, arginine 164 to aspartate 179. Another, the loop between the beta 3 and beta 4 strands, cysteine 238 to threonine 243. And the last one, the loop between beta 5 strand and the alpha 11 helix, which is expanded from LR 264 to serine 275. So we have picked up from our study, we optimized and picked up few of these loops which are very different and representative. So we are going for experimental study for them. I will just talk about one small example where you see there is only insertion of one amino acid, and because it is glycine, and what effect it is giving. So when we model this and go for a simulation study, you see that only with one glycine insertion, there is a marked change in the flexibility of the loop, and that create differentiation for the identification of the drugs which are commonly used as antibiotic. In summary, the insertion is a unique mutation to class A beta-lactamases recently observed in KPC. Though identified in several clinical isolates, further study was not performed when we have identified them. The most critical component in this insertion could be spread over the community at any time through a gene transfer mechanism I have talked earlier. Our initial study provides an interesting result, and we have taken an initiative where we have like invested a small group of already 3 PhD and 1 master student who are taking this computational analysis and doing experiment to further characterize all those variants and what type of change they are talking about. So this brings me to the experimental work. So as I told, based on our computational study based on the programming and further analysis through modeling, docking, simulation, and patterning, we have classified those class A beta-lactamases into different representatives. Class 1 or group 1 is classic beta-lactamases, where we have given 10, which is originate from Escherichia coli, and BLAST C, which is from a pathogen Mycobacterium tuberculosis. Group 2 is having PSE4, SHB, and CTXM. PSE4 is going from Pseudomonas aeruginosa, is from group 2A because it is less availability. SHB and CTXM, SHB from Klebsiella and CTXM very interestingly from an organism called Cluivera, which is not commonly found, but this gene is now coming to common organism like E. coli, Klebsiella. So this gene is coming and we have termed them as group 2B. NMC and SME are in group 3. They represent classic carbapenemases. Carbapenemases are beta-lactamases which could break the last line of defense, the last line of antibiotic, which is called carbapenem. KPC from Klebsiella pneumoniae is in group 4, and this is classic carbapenemase, but it have a number of insertion variants. GES from Acinetobacter baumannii is coming to group 5, non-classic carbapenemase. BLA from Yersinia enterolytica is a very interesting founding. It is a partial carbapenemase, and this is the first partial carbapenemase identified. This is in the group 6. The BALE and FTU, BALE is from Pseudomonas aeruginosa and FTU from Francisella tularensis. They are in group 7. They are intermediate between beta-lactamases and carbapenemase. So studying them, getting their feature help us identifying the mechanism in more detail. WAVE and SIA, they are in the minor group I have already talked about. So we have identified these 8 groups, and now we have the approach. So we have taken them and we go for cloning, overexpression, and purification. So we have taken this gene and cloned this so that we could have get the purified form of this protein. Now when we have the purified profile, we could go for individual enzyme and perform experiments to validate our result, which we have obtained through the computational studies. So first, what we have performed is called substrate kinetics. So how the antibiotics are behaving with this gene, with this enzyme, is tested through the principle. We have taken individual antibiotics, look at their structure, and because of their structure, they get a particular wavelength. Now, when the reaction happens between the drug and the enzyme, this is a very unique catalyst, a very unique enzyme which directly work on the substrate, because generally the catalysts don't work on the substrate directly, they work on the substrate indirectly. Here, this enzyme directly work with the substrate and break the 4-membered ring. The intact 4-membered ring and the broken 4-membered ring have different wavelength, which is calculated. The change of the transformation from the unbroken to the broken ring is calculated as the Kcat, the primary catalytic power of the enzyme. Here you see the catalytic power of all this BLA, FTU, EF, GES, TEM, SHB, VEL1, BLA, all are calculated with different drugs. Among them, nitrocephine, this drug is very interesting. Why? Because this drug is pale yellow in color, and in presence of beta-lactamase, it changes to bright red. We have used this change as reporter molecule and calculate the inhibitors, how they could inhibit the enzyme. How we do that? So we make a competition assay which we call inhibition kinetics, where we have added nitrosamine, which could like create this red color, but we before adding nitrosamine, we add the inhibitors like clavulanate, sulbactam, tazobactam, avibactam. So if they are going there inhibiting the enzyme, they could make the production of the red color less. So how less the red color is generated, how less the intensity is, based on that we could calculate the inhibitory constant, which is called the Ki. As I was talking about the KPC variant, so here this KPC2 is the wild type. We have taken this 113 where I have shown the result of simulation, the 44 which is having a 15 amino acid loop, and KPC 107 which is having a loop of 28 amino acid plus 18 amino acid, so 46 amino acid. We have calculated there Kcat the catalytic efficiency and Km how good they're binding. From there we could have seen that though the wild type is very active, the activity become less in the variants. Now how this reduction in activity would be used for further resistance development would come out through further detailing study. But what we have done, we have made a temperature study. Why we have performed a temperature study? Because our assumption was with this increased loop, they are like this enzyme would be affected, this enzyme's activity would be affected if we increase the kinetics of the medium. So what we did the same study with higher temperature, and you could see that at 25 it is having 1.98 and this is having 0.3. But when you go to 40, like up to 35, you see that the activity nearly becoming zero. So this enzyme, the variant enzyme, though it is showing activity in lower temperature with the increase, so KPC 107, it shows decent amount of activity, but differentiation appears with the increasing kinetic energy of the medium of the solvent. Then we go for differential scanning calorimetry. This is helping us measuring the stability of the 3D structure, the fold of the protein. So we have calculated the stability of different proteins using differential scanning calorimetry. Then we develop a fluorescence microscopy based testing, which is telling us when we have this gene, the beta-lactamase gene, we put it on a model system. Is the bacteria getting resistant to a particular drug or not? If you see the green color converted to red color in the fluorescence, that means the cells are dying, which means the drug could kill the bacteria. Whereas when the like before treatment and after treatment both are showing green fluorescence, that means the gene have provided resistance to the bacteria when the particular antibiotic is applied. We further go for circular dichroism spectroscopy. This spectroscopy gives us the characteristic fold of the protein with the secondary structure. So here you could increase the temperature and see how the temperature is altered. You might have a question in mind that don't you do the same experiment using DSC? Yes, we have done the same experiment, but that was targeting the overall 3D structure. When you do the CD, you are only targeting the secondary structure, which is alpha helix, beta sheet, and the loops. We go for temperature rate profiling, which I have shown. So here with increasing temperature, how the rate of reaction of the enzyme is altered. Why this is very important? Because now you see that global, like increase in temperature, global warming is happening. So is this global warming affecting a protein? If we have the standard data, then only we could, in the database, we could compare it with the upcoming difference created in a evoluted bacteria. We also do the same experiment with pH rate profiling, denaturant rate profiling and all. Then we come to a very interesting study which is called interaction profile of beta-lactamase with the substrates and inhibitors, which means this is the study between the enzyme and drug. So here we are like having the enzyme and then we add the drug. When the interaction happen, there would be the release of the heat. Based on the release of the heat, we calculate factors which are N. N is the stoichiometry, K is the association constant, del H is the change of the enthalpy, and del S is the change of the entropy. So we calculate nearly all the thermodynamically important factor concerning the protein and the interaction of the drug. We have also performed fluorescence spectroscopy. In fluorescence spectroscopy, we go through the protein drug interaction targeting these tryptophans. These tryptophan are natural fluorophores. Based on their changes, we calculate the binding constant, how it is binding, how it is affecting the interaction between the protein and the drug. Then we come to a very interesting study. Remember, I talked about that the enzyme we are having is a unique family of enzyme which works with the drug in a covalent way. So we utilize that to use a multi-top, a mass spectrophotometer, where we put our protein and we apply laser. So when you have your protein and you apply laser, the protein will be going through this. Now, if the protein is bound to the enzyme covalently, it could also carry with this. So you will get the mass of the protein added with the mass of the remaining substrate. But if the substrate is non-covalently bound, then when the protein would be fired with the laser, it will be denatured and the enzyme had the 3D fold, the drug was inside it, now it will come out. So based on that, we could track, how the reaction is happening, how the, like catalysis is, like catalytic chemistry is going on, what are the substrate which are remaining, and you could go into very detail of the reaction mechanism. Here you see that we are identifying the intermediates which are very fruitful for understanding the reaction mechanism, further designing the drugs. Another machine which is very unique for our use is Raman crystallography. So Raman crystallography machine is basically a confocal microscope. What are the difference here? Instead of the normal light, we put the Raman spectra. The Raman monochromator is fed and there is a very high resolution CCD. The CCD is the mostly, like valued part of the instrument. It costs like 70 to 75 lakhs. This is very sensitive. This could trap the spectra at a like timeline of femtosecond. So what we do, we try to change this setup of the normal where we put the sample and look at it. We change it and put a crystal tray. The crystal is a 3D molecule, and by setting the pinhole, we just measure the width. So why it is very important? So you have the crystal and then you add the small molecule. The small molecule have its own Raman spectra, which would be calculated before, and then all the changes happening per femtosecond would be collected by the CCD camera and compare with the already existing spectra of the untreated molecule. So if there is any minute change happen, there would be a change of the spectra, and that change would be calculated by applying quantum mechanics. So this is an example where we are using the drugs which are called mechanism based drug. There are three mechanism based drugs: sulbactam, tazobactam, and clavulanate. They are very similar to look at, and people used to consider that all their effects are similar. But through this study, we have differentiate their time of reaction. We have differentiate between their wild type and mutant, their response to the reaction and all. So this helps us or give us opportunity to go into the very deep mechanism of the reaction on a level of, on a time scale of femtosecond. Last but not the least, the technique we use is the crystallography. So this is the macromolecular crystallography unit in IIT Roorkee. These are the tray. We purify the protein and set the protein to form crystal. If we are lucky, we get crystals. These are the crystals of different beta-lactamases, NMC, BLA-C, BLA-A, SME, BEL. Then first we solve their structure to get the protein without having any substrate. Then we take the crystals and soak it with different substrates, and these are the apoprotein electron density, whereas these are the electron density of the substrates. We get the substrates inside the protein to get into a deeper look of the how they are performing the catalysis. We could understand the mechanism into very detailed. So how we use them? We use them to design novel therapeutics using virtual screening. We are developing a program with the company Schrodinger, who have a wonderful drug pipeline which is considered best in the world. We have already come up with novel penims, boronates, which are transition state inhibitors, new mechanism based inhibitors, new carbapenims with newer activities, non-beta lactams. So mostly they are beta lactams, but we have now come up with molecules which are not beta lactams. So all these therapeutic molecule we develop, and more importantly, we work with drug resistant variants. What are drug resistant variants? Which are clinical isolates. So a lot of time in different hospitals, patients are admitted where the drug is not working. So now we are collaborating with 10 different AIMS and many other bigger institutes. We are already collaborating with more than 30 hospitals, and from all those hospitals we have developed pipelines from where a new bacteria is identified. By new I mean where the drug is not working. They identify that, and when they identify that, we go through this. First, the patient samples show resistance. We go for antibiotic susceptibility testing. We detect the organism. Then we isolate the total DNA. We go for the genome sequencing and analysis. We identify the AMR gene. Then we identify the mutation where the change of chemistry in the amino acid happened. We do the 3D modeling of the enzyme. We do the docking with the substrates as I have discussed. We go for the MD simulation to understand how this resistance is working. And if it is important, then we go for further cloning, overexpression, purification, the experimental procedures, followed by the data collection with biophysical, biochemical, kinetic, and crystallography method, as I have described earlier. I would finish with a case study where we have picked out and worked with such some clinical isolates. So one of the very important part of the beta-lactamase is SDN loop. S is the amino acid serine, D is aspartic acid, and N is asparagine. So if you look, all of them, all of the organisms having this SDN loop, whereas Mycobacterium tuberculosis and Mycobacterium bovis have a G, the glycine, instead of asparagine, and this makes a significant, contribution towards the life cycle of tuberculosis. And as you all know, tuberculosis is a very important, very critical target. We wanted to investigate this. Why it is important? TAME, SHB, GES could have shown to develop resistance against these mechanism-based inhibitors. These are the mechanism-based inhibitors I have already talked about, the clavulanate, sulbactam, and tazobactam. So why they are mechanism-based inhibitor? Because for normal inhibitors, they only target the serine 70 of the enzyme, whereas this one, they deal with a second serine, which is serine 130, part of this SDN loop. So if you could change this serine, you could make your enzyme resistant to these drugs. Interestingly, all the beta-lactamases from E. coli, Klebsiella, they have already created resistance which were isolated in the patients. But there is no evidence of such resistant variants developed in Blasi, which is from tuberculosis. Our inquiry was, we all know tuberculosis is the king of this business. They're the champion. So why they don't come up with this resistance mechanism? Why they could not develop resistance? So to understand, first we did the modeling, docking and all, and followed by a comparative MD simulation. In that, we take the beta-lactamase gene from Escherichia coli, which is TAME, having SDN loop, and from Blasi, the tuberculosis, having a SDG loop. And we have performed a 100 nanosecond simulation at 300 Kelvin. We use Gromax 4.5.4 version using Amber 99 SB ILDN force field. We have used TIP 3P water model and equilibrated with 50,000 steps. So when we did that, I don't go into detailed analysis to confuse you, but we clearly see that the small loop, which is very consistent in the E. coli, you see as the black curve, is now become flexible in the Blasi when it is having SDG loop. So we understand that there is some change happening. So what we have established through computational study, we want to establish it through experiment. So our objective was, we want to make mutants. We develop S130G, which is the clinical isolates showing in other beta-lactamases. As this is not happening in tuberculosis, we by ourselves create that mutation in the laboratory. In addition, we make an alanine instead of glycine in the same position. We want to validate the hypothesis of the drug resistance. If they are changed, is it possible that they will develop drug resistance? And then we will go to the stability of the mutants, because one understanding we are having that there might be a change in the stability. So we'll test the stability of the mutants, and if we see there is a change in the stability, we will try to make a high resolution structure which will show what is the thing happening there. With those objectives, we have created the mutants. This is the wild type and this is the two variants. And when we have created them, we clearly see a change. From 1 liter, the production of wild type is 16.54 milligram. This wild type protein, whereas for glycine mutant, it is strikingly lower to 2.69 mg, and in case of S130A mutant, it is 5.92 mg. So then we tested with the drugs clavulanate, sulbactam, and tazobactam, and we get activity, as I told, inhibition in case of wild type, whereas we don't see any inhibition in case of S130G and S130A, which means all the drugs are now having antibiotic resistance. So then we go to prove our hypothesis from the enzyme to the total molecule system. We take the E. coli, and when we treated the E. coli with the drug sulbactam, in normal situation, we see the bacteria is died. You see the small pricks, like it is like this is the bacterial cell, a needle is pricked and the material came out. So this is the same picture through which we have seen it. Then what we did, we incorporate the gene which causes resistance, the gene created by us. And if you see in this same picture, you see that no change is observed. This is not dying. The bacteria is live, which means the bacteria is resistant to this drug. We did it also in gram-positive bacteria, Staphylo, and you see that there is killing of the drug, whereas when we use the resistant gene, the 3D structure of the bacteria is totally intact. Now we want to look at the stability of the enzyme, and to do that we are going to the CD spectra. These are the CD spectra of the enzymes at 25 degrees centigrade. But then we start changing the temperature to understand what is the temperature where it loses its stability. And to our surprise, but I will not say surprise, we have estimated that, but we come up with the fact that the stability, which is 64 degrees centigrade for wild type, is decreased markedly 15 degree to the alanine mutation, whereas in case of glycine mutants, it have a whooping decrease of 25 degree. So with just one mutation, the stability of the enzyme decreased 25 degrees centigrade. So then we try to understand it in the macromolecular level. We further purified the protein using size exclusion chromatography, which is needed to perform X-ray crystallography. We get the crystal and we have solved the structure. And if you look at, there is a bend in the main chain. So this is very rare, because generally with point mutation, you don't see much changes in the structure. And interestingly, this part we could not see with computational biology, with our computational analysis. So here two things. One, we understand that there is a major change happened in the backbone, which is the cause of the decrease in the stability of the enzyme. And we also have to understand that whenever we do computation, there is a need to validate, because there are rules. Based on the rules, we will do predictions. But if we don't have enough rule, if the thing we are studying is very novel, the prediction might be wrong. So we have learned these two learning through this. So in summary, SDN is a conserved loop in various organisms except for Mycobacterium tuberculosis, which has glycine 132 in replacement for the conserved asparagine. Asparagine 132 to glycine replacement provide Mycobacterium tuberculosis with higher flexibility in the SDN loop, as can be concluded from the comparative dynamic study of the SDN and SDG loop using molecular dynamic simulation. The smaller Tm melting temperature of the mutant protein supports that point mutation in Mycobacterium tuberculosis confer the enzyme instability. The high resolution crystal structure clearly shown a significant change in the enzyme's backbone, an example not available in common places. Overall, this explains the molecular reason behind the occurrence of a natural phenomenon, which further explains why the S130G variant is absent in the clinical isolates. So coming to the conclusion, we have opened a Pandora box of one of the most interesting and demanding clinical problem of the current era, the antibiotic resistance. The computational part of our study, including programming, modeling, docking, simulation, AI-based patterning, is instrumental in covering the total space of the problem, because the total space is very big and it helps us strategize our experimental studies. The experimental studies not only validate these concepts but also help in understanding the subtle differences between the beta-lactamases of different subclasses, natural variants, as well as enzyme-drug interactions. This understanding would also be extremely helpful in designing new generation theranostic, especially in testing newly invented drugs. So this is, if this is the common thing, now coming to a philosophical understanding, we could not stop AMR, but we have to slow it down to get some time every time so that we could claim our existence. And it is high time so that we could understand that it is not possible for individualistic lab to work. It needs the clinicians, to the clinical microbiologists, to the molecular biologists, structural biologists, computational biologists, AI specialists, device developer, therapeutic development, pharmaceutics, all to come into a common platform to come up with a overall solution. Very fortunately, the government have taken an initiative which is called One Health Compassion against the AMR. So coming to the students who have contributed, Vivek and Khalid, they work with the Param Ganga supercomputing. They do all the computational work. Sorjo, Kunal and Harshita, they are working on class A carbaphenemases. Ria, Tanmoy and Abir, they are working on like APC, the insertions and some of the novel beta-lactamases. Sumecha and Jyoti, they are working on metallo-beta-lactamases. Nitish is working on environmental AMR, developing devices which are helping to identify AMR in environmental and medical condition. Deepika is working in collaboration to develop nanoparticles, the nanoparticles which are being doped and create power to further treating the biofilm and all. Advaita is working on brain-based signaling, which is used for diagnostics. Anish was an alumni of IIT Kharagpur. He worked with me for around 2.5 years, and he worked as an intern, but he was more than an intern. He guided the whole process, the huge team of intern and all. We have fortunately found a lot of funding from SERB, ICMR, DBT BIRAC, Spark UK, DHR, BRNS, IWMI, UCOST, Royal Academy of Engineering, DST, Dibya Sampark, iHub, IIT Roorkee. But I will just mention three things. Recently, we got a very big grant on ICMR where we are working with 15 institutes to work on leukemia cancer, where the cancer patient is affected with antimicrobial resistance. One of the very new area I'm working where we are found like getting funded with IWMI UMI, which is an international organization working on water and environment. First time they introduce AMR modeling, and especially in the developing and underdeveloped countries, we are involved in the first ever modeling of the river or surface water, where we take different factors which influence the AMR. Dibba Sampak they have invested 1 crore with us and push us to develop the first company, which is Doer Healthcare Private Limited, which is now working towards developing devices which will be working in different level of diagnostics for AMR, the level of AMR, or the drugs, the antibiotics which are could be identified and all. This is the team of our group, and I will just end up with a philosophic note. AMR, we are now talking about, India is prospering and all, but AMR like is an indication of the country's development. As Darwin told, if the misery of our poor is caused not by the laws of nature but by our institutions, great is our sin. So we should have taken very good measure so that we could control AMR in the basic level, so that the below poverty level kids who are growing up would not only be deprived by nutrition but also getting diseases, chronic diseases, which is because of the pathogenic organism around. And I will end up with a quotation from poet Sukanta Bhattacharya. I want to make, which is a promise from myself also, I want to make this world healthy and ever grown. This is my soulful promise to the next generation newborn. এ পৃথিবীকে এ শিশুর বাসযোগ্য করে যাব আমি, নবজাতকের কাছে এ আমার দৃঢ় অঙ্গীকার। থ্যাংক ইউ।
Thank you, sir, for your valuable talk. If anybody want to ask any question from sir, so they can ask.
Hello.
Yes.
Professor Hazra, it was nice and informative talk. I have one query.
Sure.
In the development of your pipeline for the development of data set, you mentioned you curated your data from various literature sources and you consider some other information as well. And also you mentioned you have one headers in your curated data, Hazra
We have.
headers. You have header specific to
Yes,
some.
yes.
Do you just elaborate that headers?
Yes, actually, very good question. Not a lot of people ask them like ask this question. So, what is our target? Is like as you told, some of them we have collected from the literature. Some of them are created by our in silico analysis, right? And then some of the work will come from different level of experiment. So, for one enzyme, there would be several different data, right? So we make the headers in such a way that later with one search, you get all the data regarding one enzyme. That's why when we are creating the headers, we are putting tag so that in one call, individual enzyme, all the different set of data would be coming together. You don't have to go into different level to see the data for one enzyme. That's what we are looking for.
Uh, uh, is it already implemented in your pipeline so it can automatically fetch the data?
Yes, but the thing is, as I told, we are now having the like gene sequence, protein sequence, and we have created the protein structure, like interaction dynamics and network. Beside that, we have this, kinetics, structure, fluorescence data, binding data, and all these. So biology data. So those are not included. We are on the process, but which we have done are included there.
Thank you.
Thank you very much.
Hello, sir. Am I audible?
Yes.
Yeah. Thank you, sir, for the wonderful lecture. Sir, I am an undergraduate student. So, sir, I have two like basic queries if you can kindly answer.
Sure.
So the first question is, so you mentioned the use of Autodoc Vina as your docking tool. So can you just compare between this Autodoc Vina and Autodoc? Because I have seen people read in the literature, people doing this high throughput virtual screening using Autodoc Vina,
Right.
and then with the selected
You
candidates
mean
based
Pyrex?
Pyrex or batch docking, even
Yeah.
the loop, using the loop mode on
Correct. Correct.
this Autodoc Vina, and then coming up with the best few candidates, and then for greater conformational sampling, they are using Autodoc for the
Right.
subsequent part. So, is this approach correct, or is it like not correct to combine these two forms of docking together?
I think it is more important. Like what I think, uh, like both the Autodoc and Autodoc Vina are very widely used, right?
Yes, sir. Yes, sir.
So they are validated. The more important thing when I consider automation is the personalization of the active site and all where the binding happens. There are a lot of automated program and many of them computationally are very good. But if you look at the broader perspective, you are not performing docking to perform docking. You are performing docking to get a proper protein drug interaction profile, right?
Yes, sir. Yes, sir.
So that's why we are picking up this personalized program where, you know, like we will publish this later also, where you get an enzyme, then you go for the understanding of the enzyme, how the enzyme do catalysis, and you mention those residues. So here, that's the difference we are doing. We take their program and what we did, what change we made, you could have identified the amino acids which are important and mention them in the grid setting and all.
Okay. Yeah.
Yeah.
Thank you so much, sir. Thank you. Thank you, sir.
Hello, sir.
Hello.
So it was a very nice and informative talk.
Thank you.
So my question is that, sir, you said that you have performed like docking, sir, like
Yes.
for that proteins. Like I am asking, like if the docking was like blind docking, or it was like you have
No, no,
identified
like
the.
that's what I answered. So it's not blind docking because we know about these enzymes. Like my since my postdoc, I am working on this beta lactamase project, and that time I was working only on tuberculosis. I get this advantage of coming back here and like in US they have restriction like they work on tuberculosis or any one organism. Here, I don't work kind of as a biologist. I work like a chemist. To me, the importance is the interaction. So instead of being restricted to one like organism like tuberculosis or Klebsiella, I am focusing on the chemistry of those enzymes and we put options so that every time you come up with a new enzyme, you like new beta lactamase, you could mention the important residues and regions, volume, active site volume and all so that it could be very personalized.
And sir, one more question also, like
Yeah.
like for when we perform docking, like we get many poses, like
Yes.
so like sir, how many
Excellent question.
run
Excellent question.
how many runs like sir it is required to like identify the structure that have the like the lowest binding, like in binding energy is the most, like.
Yes. So again, as I am a chemist in background who understand biology, my approach of computation is from another angle from where you are looking at. You are looking at through optimization of the technical procedure, right?
No, I am saying, sir. Yes,
No,
sir. Like
that is technique, right? That is
yes,
not
yes,
involving any experimental data. But me being a crystallographer, I know where the actual compounds are having like developed their interaction in a experimental data. You understand what I'm saying?
yes, sir. Yes,
So beta lactamase is a mostly studied enzyme.
yes,
It is not the scenario that the docking we are doing, most of them are not present. Some of them are already present in the literature as a experimental data. We pick them up, we use those conformation as standard conformation, and then compare with the newly docked poses, newly docked complexes. Is it becoming clear what I'm saying?
yes, sir. Yes.
Okay.
Thank you,
So it's
sir.
a, yeah, we are combining the theoretical approach with the already existing experimental data.
Hello, sir. I am Jishnu. I am an undergraduate student at IIT
Hi.
Kharagpur. I really enjoyed your talk on AMR, and
Thanks.
I have a question for you.
Yeah.
So, I am working on a protein called carbonic anhydrase, which is
Uh-huh.
but because it has zinc in its catalytic site. Now,
Correct.
I'm having trouble with my docking experiments. That is when I'm running my dock using the autodock for zinc parameter file. I'm doing a redocking experiment first, and my redocked pose is not still data available.
Yes.
So any
Yes. But
suggestions for
yeah,
that?
no. It does not need suggestion. There is a information that in in Autodoc is the platform in UCSF, right? So they like they not only work there as a group, they are open to many other people. Like today, you could develop a method and you could submit to Autodoc platform. So similarly, there are already done work where they have taken zinc and developed parameterization. You just download. Hello, hello.
Hello, yes, sir.
Yeah. So there is already people done zinc parameterization. You just search zinc parameterization and you will find it. So you
Yes,
have
sir.
to
I'm using that parameterization.
okay,
That's what I'm
okay,
saying. After using that, my docked pose is not matching with my crystal data available. So in my redocking experiment, so
okay.
that is where I'm stuck.
So you mean what you mean is you already have a exemplary experimental information of carbonic anhydrase, and now when you are doing the same thing, you are not getting that close to.
Yes, sir.
Like don't discuss it here. I will, if you want, I could take a look, and this should be like I have to look at the particular thing happening there.
All right, sir. I
Yeah,
can mail you maybe later. Thank you.
sure, no problem.
Hello.
Hi.
Good morning, sir. Am I audible?
Sure.
Yeah. Thank you for such a lovely talk on the AMR, sir. It was really nice hearing about this. The work you have done is amazing, the database and the creation. Sir, I have a question regarding the sequences when you align the 25,000 sequences and later on filter them towards 1916 sequences. You mentioned that
Right.
you cluster the sequences. So I wanted
No,
to
we
know whether.
cluster after getting into 1916. So first we do the reduction because
Okay.
you get same sequence. People
Mm.
have submitted anything. That's the problem in biology. A
Yes.
veterinarian in one institute get a new bacteria, sequence it, and anyway submit the data.
Mm.
Now the softwares are doing all this comparison or all, but still they report them through publication. So that's what the problem is. Same sequence with different name, same sequence with partial thing. So we tried to filter those out. And there we get 1916 sequences. Yes.
Yes, sir. So they were later clusters. So actually, I'm also applying the similar thing on few of my sequences.
Uh-huh.
So because it's the families, obviously, the sequences are very redundant. So I try to sequence them and everything. But I just wanted to know whether the supervised machine learning is good for that, or the unsupervised method that you use for the clustering, or whether the specific sets that you applied for the clustering method.
Good question. I think like I am also not sure about honestly. Oh, we perform many, okay,
Okay.
and see where the optimization happened. The good thing is we know most of the important parts, so we have some features in our mind. When those mate, we go for like we optimize that process.
Okay, so the known features are always a good way to go with the clustering.
Always like honestly
Yes.
in this session uh, like Dr. Mitra is there are a lot of computational people. I am not a strongest computer people to look at honestly. My strength is to combine the existing method like techniques with the experiments which I understand very well,
Okay.
and definitely the chemistry, because having a chemistry background I understand the chemistry of the sequence enzyme and all. So though that is strength from my side. But recently we are actually collaborating with a new faculty joined with us, Professor Devesh Bhimsaria, who is a AI expert, and he is optimizing them. Like probably after some time I could talk about when he will come up with the first set of results. But for me, I do trial and error.
Okay. Well, thank you so much, sir. That was a lovely talk, and it's a good
Thank
wish.
you.
Hello, sir.
Yeah.
Uh, Thank you for your wonderful lecture, and it was very informative. Sir, I have two questions.
Sure.
First of all, sir, the KPC2 variant and KPC117 variant, you have shown the efficiency difference. Sir,
Uh-huh.
due to this field, I do not know how to calculate or measure these efficiencies. Can you please tell me how? How have you done this?
Yeah, I tried to talk about when I was talking about the substrate kinetics. So these calculations are not general. Okay. These calculations are individualistic. What I mean, if you go to look at, can you see this compound?
Yes.
So you see that there is an intact four-membered ring
Yes.
and there is a broken four-membered ring. So this four-membered ring is broken by the enzyme. So the intact four-membered ring compound and the broken four-membered ring compound are showing their peak into different wavelength.
And
Okay.
from yes.
Okay, let's think that these compound have a particular wavelength to see their peak depending on the concentration of the molecule. You could understand that.
Yes.
This will be lost when the molecule lose their four-membered ring.
Yes.
We measure this rate of loss. So we get kcat based on the measurement of the rate of loss of this like compound. You have a peak and the peak get reduced. We measure the rate of that loss.
Okay. From there, you are calculating the
Kcat.
efficiency.
Kcat.
Okay.
Okay.
Okay. Okay.
For Km, it is relative because there is no reaction for the inhibitors. So there we use this reporter molecule nitrosaphine. Normally this molecule have this pale yellow color. It turns into a bright red. Okay.
Okay.
So for a certain concentration of the molecule with a saturation concentration, we add a certain concentration of the enzyme and get the rate, but now not with the change of the wavelength, but by change of the color from pale yellow to red. Is it possible?
Yes.
Yes. Now, if you have an inhibitor that will inhibit this nitrosaphine to work because there will be a competition.
Yes.
So the red color generation would be reduced.
Okay.
And based on the reduction rate, we calculate the Ki, the inhibitory constant.
Okay. Okay.
Actually,
Understood.
what you asked is a very good question, because we have to understand how they are working. There is definitely enzyme kinetics, but it applied to different drugs, different enzymes in a totally different way. Some are causes change in the wavelength, some cause change in the color. And here you might think that, sir, if there is no such characteristics, then how we calculate the rate for that enzyme, right?
Yes. So
We,
you
we,
have taken
we
the
need
same
to,
procedure to check the temperature-based studies as well when you have
yes,
calculated the
yes.
efficiency.
No, but what I'm saying is, let's say you have an enzyme which does not have any reporter molecule which change the color or change the wavelength. How you calculate the rate? Because as a biochemist, we develop assays also. So in that case, you will generate a secondary assay, which means you have to add another enzyme which is standard, another substrate which is standard. But that reaction will get one substrate from the first reactions like portfolio. So you actually measure the second enzyme, which is standard, but how the first enzyme's reaction product is coming, you will calculate that rate to calculate the catalytic activity of that enzyme. So in that way, it is not applicable particularly to everyone. Every enzyme have its own set of assay you have to consider.
Okay. Sir, my next question is from Raman crystallography slide.
Uh-huh.
There you have said that we can get different crystallographic measurements from
No. We have a crystal. Here, crystal is the platform.
yes,
Okay. So, do you have any idea about confocal?
yes, sir. I
So,
use confocal microscopy.
fantastic. So, confocal measures Z. If you focus the pinhole, it will only measure the Z, right?
Yes,
So, we focus the crystal on the pinhole so that it only measures the inside of the crystal, the width of the crystal.
okay,
Now, it will not measure the drug when it is outside. It will only measure the drug if it goes inside.
yes,
So here the like good part is, if you have to measure it with solution, the concentration of the molecule is much less in a solution than in a crystal.
yes,
It is 10 to the power 12 to 10 to the power 16 times more concentrated. So basically, if I try again, so you know confocal, right?
yes, sir.
In confocal, you use light microscope.
Yes,
Instead of light, you use Raman microscope. Like, just change the spectra from light to Raman source. Okay.
yes,
Still you could measure it.
yes,
Then why I need a Raman microscope, like Raman crystallography? Because in case of protein, if you keep the protein in solution, you don't get Raman spectra in a detectable amount.
because Raman signals are weak.
Fantastic. And that's why, being a you don't see Raman is very, like sensitive instrument, but still we don't do much experiment with Raman when it concerns to the protein, because of the faint signal we are getting. Why? Because the alternative is resonance Raman or surface enhanced Raman, right?
Yes,
But when you apply resonance Raman like laser or surface enhancement, there is a high chance the protein would be denatured.
yes,
That power of laser we are replacing by replace it with a crystal where the protein is much more concentrated. That's the key of the Raman crystallography. Unfortunately, this time no one in the world used it. When I was learning, William Perry was the innovator. A lot of people, like 20 people, used to do it in the world. Now it left to only me after William Perry, retired and all. So it's a very interesting, unique instrumentation.
Yes, sir. Sir, when I use Raman, basically I use 532 nanometer laser. But the thing is,
Yes,
ki I get a lot of noises. Can you please suggest
exactly.
anything where I can remove these noises? Or
Exactly. That's how that we don't know how to remove it. That's why we go for a different, we are a crystallographer, so we know how to grow crystals. We use it to merge two things in a technique.
okay, okay. Thank you, sir.
Yeah.
Thank you, sir.
Thank you. Thank you very much. Lots of questions. I really enjoy.
Okay, sir.
Okay, so thank you, Dr. Hazra. It was a nice talk. Okay. So now we will move on to our next.
Thank you.
We'll move on to our next.
I extend a special welcome to our distinguished guest, Professor Debulai Chakravarti. We honored to have you, sir, here with us. He is a distinguished figure in the Department of Metallurgical and Materials Engineering and currently serving as the Associate Dean of R&D, IIT Kharagpur. His academic journey is marked by dedication to excellence in both research and education. His pursuit of knowledge led him to earn his PhD from University of Birmingham, UK, and he was the recipient of the prestigious Overseas Research Scholarship. His contribution to the field of metallurgy and material engineering have been widely recognized, as evidenced by his recipient of the Metallurgist of the Year award in 2020. Moreover, his leadership and vision have been instrumental in steering our institution towards excellence in both research and education. Now, I would like to call Professor Debula Chakravarti sir to have some words with us.
Thank you for the nice introduction. And I am really glad to attend one more workshop in the center, CCDS. And I'm very impressed with Professor Shomodi, Professor Shomnath, Professor Praloida, and their team, that with the enthusiasm with which they are organizing this kind of workshops very frequently. And yesterday only I was attending some other workshop where our Dean Student Affairs, Professor Bhargav Mitra, was there. So he was telling that these kind of workshops can be more attractive mode of knowledge transfer nowadays, because students are more reluctant to attend classes, typical classroom teaching. So this is also a way of knowledge sharing, which may be more and more coming as play to come as an important role. So and I am very great that the CCTS is doing this. Some of you, I heard that you have come before also in this campus, those who are coming from outside. You know that Kharagpur IIT journey started in 1951, and 18th August is our foundation day in the place of Hijli detention camp. So this is one of the possibly the only reputed institute which is situated on a prison house. So if you get time, just have a look in our old administrative building where some prison cells are already there. And from a very humble beginning, now we have more than 16,000 students, 800 faculty, and about 1,000 staff members. And me coming from SRIC, we manage projects. So all the project related activities, funded projects. So we hope that you will share your knowledge and help this center to grow, so that more funded projects and more challenging problems come to us, and our very much able faculty members, they are there to address those. So this is high performance computing on bioinformatics. I think this is a very area of current interest. So it's wonderful, and I heartily welcome you all to this beautiful campus. Have a very nice workshop in the free time. Have a roam around the campus and see our vast campus and enjoy, and very living campus, lively campus we have. Night also you will find students roaming around in the roads. So enjoy your stay. Thank you. And I wish all the best and success to this workshop.
Sir, Soumyajit De sir to present a bouquet to Devolai sir. Now we will go for short break of 10 minutes. We will be back in 10 minutes. Have a tea and breakfast. And for online use participants, be there in 10 minutes. We will starting soon.